Hello, my name is Ernst Schmid, and I'm excited to give you this short introduction to the Predictome, an online resource I created while a graduate student in the Walter Lab at Harvard Medical School. The Predictome is a database of predicted protein-protein interactions that we identified using an in silico screening pipeline based on the structural prediction program AlphaFold. The human genome encodes 20,000 canonical proteins, most of which are thought to function by interacting with other proteins through protein-protein interactions, also known as PPIs. Current estimates suggest that there are several 100,000 functionally relevant PPIs, but most of these are unknown, and only about 10,000 human PPIs have been structurally resolved in the PDB. Filling this gap in our knowledge of PPIs is critical to fully understand human cell physiology and to fuel drug discovery. AlphaFold represents an exciting opportunity to help fill this gap. AlphaFold is a deep neural network that predicts the structure of a protein or protein complex based on amino acid sequences. Importantly, it also reports its confidence in the prediction it makes. Several groups, including ours, realize that AlphaFold can be used not only to predict the structure of known complexes, but also to discover new protein-protein interactions. In this approach, a bait protein is folded with many prey proteins, and the hits are ranked by AlphaFold confidence metrics. By combining such in silico screening with structure-function analysis, we and others have identified a handful of new PPIs that have revealed new mechanistic insights into diverse processes. An obvious next step is to fold all 20,000 human proteins with one another to try to uncover all possible PPIs. However, that amounts to 200 million binary combinations, which would require tens of millions of dollars worth of cloud computing to model with AlphaFold. To overcome this challenge, we first needed to develop a triage step to help identify the subset of most likely interacting pairs. To this end, we trained a new machine learning classifier called Kirk that rapidly assesses whether two proteins interact by considering their biological properties such as co-expression, co-localization, and genetic codependence. After calculating the Kirk scores for all 200 million pairs, we used AlphaFold Multimer to fold the top 1.6 million pairs. This took 3 months on a 256 GPU DGX cluster generously donated to us by NVIDIA. We folded each protein pair in 3 out of the 5 uniquely trained AlphaFold models, so there are 3 predictions for each protein pair in our database. We then used a second classifier called SPOC, Kirk, SPOC, get it, to make a final judgment about whether each predicted protein pair represents a functional PPI. Like Kirk, SPOC considers biological properties, but it also examines structural features of the protein pair. These include AlphaFold's confidence metrics along with measurable properties of the interface such as the amount of buried surface area. By benchmarking SPOC against known positive and negative PPIs, we calculated the likelihood that a prediction is incorrect at any SPOC score. This is known as the false discovery rate, or FDR. For example, pairs with a SPOC score above 0.86 have a 10% chance of being incorrect. In the end, 16,000 pairs emerged from our pipeline with SPOC scores greater than 0.86, and about 5,000 of these are not supported by prior evidence, indicating that they are novel. If we drop the SPOC score to 0.5, the yield of PPIs increases to 113,000, but the false discovery rate also increases to 50%. Now let's look at predictomes.org, where you can interactively browse the data for the 1.6 million protein pairs we modeled as part of our screen. On predictomes.org, you can find a link to the human predictome under the data menu at the top of the main page. Once at the human predictome, you can navigate to your favorite protein via the primary search bar. To demonstrate, I will pull up the data for one of my favorite proteins, the replication factor Donsin. Pressing enter will retrieve a list of potential Donsin partners that emerged from our pipeline, ranked by SPOC score high to low. Please note that because we only ran AlphaFold Multimer for high Kirk scoring pairs that were under 3,600 amino acid lengths in total, you may not see every pair you expect for your protein of interest. In other words, while our screen is expansive, it is by no means complete. Moving back to Donsin, Hits that earn a SPOC score of at least 0.86 are highlighted in blue. The table lists other confidence metrics, such as average models, which quantifies how well the 3 independently trained AlphaFold models agree on a prediction. Among pairs with the lowest SPOC scores, say 0.5, those with a good average model scores are especially worth paying attention to. As you can see, our pipeline retrieves Donson 4 known partners, which include GINS 4, MCM3, Poly 2, and TOPPP1. It also nominates new partners such as the histone chaperone NASP, which we identified via this screen and which we have now experimentally verified. Clicking on a hit, such as GINS 4, which is also known as SLD5, retrieves an information page with confidence metrics and an interactive viewer that displays the predicted structure. You can view each of the 3 predictions here, or superimpose any matching PDB structures here, and finally, you can download the prediction here by going to the bottom left and clicking the download button. We provide several additional tools to help interpret the prediction. To simplify the structure, it can be filtered by the PLDT confidence score, which is reported on a scale of 0 to 100. PLDDT is an estimate of AlphaFold's confidence in the position of each amino acid relative to its closest neighbors. You can color the structure by PLDT score, which reveals the regions predicted to be disordered have PLDT scores below 50 and are colored orange, whereas the residues in globular folds that are confidently predicted have PLDDT scores above 70 and are colored blue. As expected, filtering out low PLDT residues removes the disordered parts and suggests that Donson binds GINS 4 via Donson's globular fold and via an N-terminal Donson peptide. So how do we know which of these interfaces is contributing to the confident prediction? For this, another confidence metric called the predicted aligned error, or PAE, is extremely useful. The PAE is reported between every 2 residues in 2-dimensional graphs, where blue means the PAE value of the intersecting residue is near 0 angstroms, whereas red means it's 30 angstroms or above, meaning it's very uncertain. Importantly, the aligned error does not report on whether 2 residues directly interact. Rather, it's an estimate of AlphaFold's confidence in their relative position to one another. For example, if AlphaFold estimates that the distance between 2 residues is between 9 and 10 angstroms, the predicted aligned error between these 2 residues should be 1 angstrom. A block of blue within a single chain indicates a well-folded domain where AlphaFold is confident about the positions of all amino acids in the domain relative to one another. To evaluate protein-protein predictions, however, we look for low PAE values between different chains. For example, residues 1 to 25 in Donson show low PAE values relative to GINS 4. In other words, AlphaFold is confident that the N-terminus of Donson interacts with GINS, whereas the PAE is higher and therefore less confidence for the globular domain-domain interface. This page displays other useful information, such as the residue-level PLDT scores for each protein, which often reveal a massive increase in local PLDT score upon complex formation. For example, the normally consistently low PLDDT highly disordered N-terminus of Donson displays a massive spike in PLDT in the binding region when it is modeled with its correct partner GINS 4. We also display the rate of evolutionary change for each residue, with a low rate indicating high conservation. This shows that even in regions predicted to be disordered, residues that interact with other proteins, such as the N-terminus of Donson, can be highly conserved. Predictomes.org also features some of our other, more focused in silico screening datasets, such as an all-by-all interactome of 300 core genome maintenance proteins. Finally, we want to stress again that structure prediction is far from an exact science. Some PPIs with very high SPOC scores will be false, and conversely, some functional PPIs will have low SPOC scores or drop out of our pipeline altogether. When choosing actionable hits, the lower the SPOC score, the more important it is that there is some external evidence that suggests that an interaction could be real. But whatever the properties of a hit, it is important to remember that the PPI reported is a prediction, not a fact. So experimental verification is crucial. Ideally, you should use site-directed mutagenesis to verify the predicted interface and ultimately determine an experimental structure. With these caveats in mind, we hope that the Predictome dramatically accelerates the pace of your research as it has ours. Thank you for listening and happy predicting.